everyone. It is our pleasure to have you join us today. We are honored to have Dr. Avinash from Imperial College London and Dr. Chance from Duke-NUS to be our keynote speakers today. They will sharing with us about the statistical analysis, specifically on using R and Python programming. This is the agendas for today's webinar. We'll start with brief introductions to the IUBMB training initiative, followed by a talk by Dr. Avinash on R and a Q&A session right after his talk. And after that we'll have Dr. Chance to talk about Python, and followed by the second Q&A sessions. Please feel free to drop your questions at the Q&A chat box. We are the IUBMB training initiative, which aims to unite emerging scientists from around the world. Our objective are to share the scientific resources and knowledge, develop skills, increase accessibility and inclusivity, and promote sustainable science. IUBMB training initiative is composed of members from 4 different regions: the Europes, Americas, Asia, Oceania, and Africa regions. These are the members from the Asia-Oceania regions. We have Jessie, a PhD candidate from Duke-NUS Singapore. As the advisors, I'm Grace, the regional leaders for Asia-Oceania. And I'm a PhD student from the University Tunku Abdul Rahman, Malaysia. And we have Ben, a PhD candidate from University of Auckland, New Zealand. And Datri, a PhD candidate from India Institute of Science. We are glad to welcome our speaker for today, Dr. Avinash and Dr. Chance. They'll be sharing on the R and Pythons, both the very powerful statistics tools. The titles for Dr. Avinash talks for today is Peeking Inside the Black Box, Demystifying Statistical Analysis in Biology using R, and Dr. Chance will share on the topics applied bioinformatics using Python programming. Before we begin, let me give a brief introduction to our first speaker, Dr. Avinash. Dr. Avinash is a reader of the innate immunity and infections at the Imperial College London and has a satellite affiliations at the Francis Crick Institute. Dr. Avinash has developed online learning resources on learning R, biological statistics, free R package Graphify for plotting graphs and easily performing advanced analysis. In addition to teaching immunology and host pathogens biology, Dr. Avinash runs workshops on R and statistics for students at Imperial College. Without further ado, let us welcome Dr. Avinash. Dr. Avinash, over to you.
Thank you. I'm just going to share my slides. Can you see my slides?
Yeah, you can see your slide.
Get the pointer on, and then I'll be good to go. All right. Thank you very much. It's a pleasure to be here. It's always great to be invited for a talk by students. And Doritri, who's one of the trainee initiative members, was in my lab for a bit, and she's been through one of my workshops. So it's really a pleasure to be here and share some of this work with you. My day job is as an innate immunologist. We work in host and pathogen interactions. But it was during the pandemic where, like a lot of people, I was sitting twiddling thumbs, and I decided I needed to teach myself something and keep myself occupied. And that's when I got into R and RStudio, because it's open source, it's cutting edge, and it's a large worldwide community of people who have lots of learning resources made available worldwide. And I would highly recommend, if you've not done R yet, get into it. It's just really exciting, and it's very useful these days for data science as well. There are lots of resources. This is the link to the workshops I run. And even if you've never done any R before, this will take you from very basics all the way to some statistics that I'm going to talk about. But of course, it's not just the software, right? You can use any software you like, but unless you understand the statistics, the software is going to be of little use to you because you may make mistakes in the tests you use, for example. So I also, during the pandemic, wrote a little book. It's a little book online that's also, again, freely available. It has basic statistics theory written in a way that is accessible to immunologists and microbiologists, for example. But it would also be useful to any in vitro or in vivo experimental scientists worldwide. There's an R code for lots of common tests as well as power analysis, and again, available at this particular website. And then, of course, I made this particular package called Graphify that I will talk about towards the end of my talk, which integrates both data visualization as well as data analysis through modern advanced statistics in R. And as far as the overview of this talk, the one take-home message that I would like you to take with you is to plan experiments and analysis at the same time. And this will do you a lot of good in the long run. I will try and speak a little bit on data visualization and statistics as we go, but it's a short talk. So you will have access to the resources that I just shared to learn more about a lot of other aspects of R. So as PhD students or undergraduate students even, we are quite often happy that we've just done an experiment. And that's great. And you should be proud because doing an experiment is a big deal because it takes a lot of effort and learning. But then as a boss or my boss used to ask me, have you plotted the data? Have you analyzed the data? What are the data telling you? That is when a lot of us become a bit more nervous and we're like, what, there's more to do after an experiment? Because of course there is, right? And so one of the things that's useful is to plan experiments. And just like we plan controls and protocols, plan the methods and the comparisons, the tests that you would like to do at the same time. And over the next few slides, I'm going to go through some of these points about planning statistical analysis at the same time as planning your experiments. So the first question is then, which or how many comparisons should I make, right? And you'll be pleased to hear that this actually is not a question for statistics. It's a question for you as the biologist or the experimental scientist, because you may have designed a new drug, or you may have a new mutant enzyme for which you're finding the KCAT or KM. It's for you to decide what you want to compare, right? And so a lot of thought has to go into this, and this is not a statistical question. But what is a question for stats is which test to use once you've decided what comparisons are meaningful for your experiment. And right from our undergrad, I'm sure most of us would know that if you have two groups, you would use a student's t-test. But what few of us actually appreciate is students' t-tests is actually a special case of an ANOVA. And you must only use it if you have exactly two groups to compare. If you end up having three groups and you do multiple t-tests, you run the risk of having false positives, which means a falsely low p-value. So this is, for example, when you have one factor and a couple of groups, it's a simple experiment. Quite often we have more than two groups, and so it's good to think of these as ANOVAs in general. For example, you may have a control and a couple of different treatments. But many experiments are even more complicated. You may have groups within groups. For example, you may have a wild type genotype, which you either treat or don't treat, or you may have males and females, or you may have different times and so on. And so these kinds of hierarchical designs are better analyzed in what are called factorial ANOVAs, which are more general cases. And one-way ANOVAs and student t-tests are more specialized cases. But what is even more interesting and something that you can do in R, which is more powerful, are what are called mixed effects. Because, for example, we are aware that quite often we would repeat an experiment where these are the groups of interest to us, these are the comparisons we want to make. But we are also aware that when we repeat experiments multiple times independently, there's also experiment-to-experiment variation. And with mixed models, you can have mixed factors, those that are of interest and those that are not of interest. And therefore, you have more power to detect any differences that you're interested in. And this is easily possible in R as compared to most other software. And so one thing to remember then is that modern ANOVAs are actually linear models. What you do in R, they're actually technically called linear models. And one way to think of linear models is basically, the simplest thing is to think of them as fitting straight lines to data, which we often do, for example, when we are plotting a standard curve for an ELISA or a Bradford in a colorimetric assay, right? And one of the things that we do there is to check if the line that we have fit is a good fit to the data. And just like that, for linear models, as well as traditional students' t-tests and ANOVAs, you must check whether the residuals of the test are approximately normally distributed, which means whether the observed quantiles and the predicted quantiles are roughly on this diagonal line. And if they're not, the model is probably not a good fit and you should not proceed with comparisons. You could consider transformations such as log or logit and so on. And quite often an easy tell is if you're plotting your data on a log axis, for example, if you're doing RT-PCR, or if you're measuring colony forming units or plaque forming units in bacteriology or virology, you probably should be transforming your data with log transformations because your data are log normally distributed, right? So with that in mind, the next question that most of us struggle with would be how many repeats or how many independent samples do we need? But to answer that, you first need to be sure you understand the difference between technical and biological or independent replicates. And so what are these? So one way to think of independent sampling is, for example, drawing a ball like in a lotto or a lucky draw from a big bag with lots of balls which have numbers on them. This is like thinking you've been asked to find the mean and the standard deviation of all the numbers in this big bag. Independent sampling would be drawing one ball, mixing the bag and taking another sample independently. Whereas a technical replicate would be then drawing a ball and then asking your friends just to read the number on the ball just to improve accuracy that you've noted down the correct number. Another way to think of replicates is to think of representative sampling. For example, an analogy from exit polling or surveys or clinical trials, for example. Are you sampling representative members of the population that you're trying to study? For example, if you measure or ask a survey to a family of pencils, for example, who are not representative, they may all say we don't like erasers, right? So you need to be aware that biologically different doesn't necessarily mean biologically or statistically independent, and avoid oversampling your similar individuals, right? And so just in terms of generalities, make sure you average your replicates, and what matters is the quality of the replicates rather than the quantity of the replicates. That still leaves us with how many replicates, now that we understand what is an independent replicate. That, again, you'd be pleased to hear, is not a statistical question. It starts from a biological question as to what is the meaningful difference in your system that you want to measure. For example, if you have an enzyme and you made a mutant that you want to compare, what would be biologically meaningful change in the Km of that enzyme? Or what would be a reduction? Let's say you have a new drug for cholesterol. How much of a reduction in cholesterol levels by this new drug would you be satisfied with? Is that 25% reduction? Is it a 50% reduction? That's a question for you as the biologist to answer. And of course, you also would need something called the noise or the signal, the standard deviation in the groups, which you can get from a pilot experiment or from literature. And one way to look at this is you need to know what the signal you're trying to measure or the difference between the two groups and the noise, because the signal to noise ratio is what you're trying to optimize in your experiments. And so power analysis is a more complex way, or more, sometimes people are afraid of this, is the way to determine sample sizes. Basically, it tells you if you have decided this is the effect you want to measure, and if it truly exists, what is the smallest number of samples you must use in an experiment to be able to reject a false null hypothesis at least 80% of the time. And so this increases reproducibility and robustness. And of course, there are great packages. G-Power is very common. It's very good. In R, you would use a package called Power or Simmer, which is simulation-based, which is even more powerful for power analysis in R. So then that brings me to, what about the p-value? What did you mean by reproducibility and robustness through power analysis? So to understand that, we need to appreciate distributions and outliers, which is something that is common to pretty much every parametric statistical test. So most of us have seen these kinds of distribution curves. These are just frequencies of the values in a particular population. Let's say we are measuring the doubling time of E. coli growing in LB, which is, let's say, about 20 minutes, with a standard deviation of 2. If that is your LB, that is your population, 95% of the values would lie within this region, which is about 2 times the SD. And we also know that about 2.5% lie in the 2 tails. And that is where this outlier idea of about 5% or P less than 0.05 comes from. We have, by convention, decided that 5% values of any distribution are the outliers, or they are in the tails of the distribution. And so what this means is, with this kind of background in probabilities, you can answer questions such as, if my E. coli or my population has a certain mean and a certain SD, in this case 20 and 2, what is the chance of obtaining a value of about 28 or so, or 28 or more from experiments? Is this a random likely chance? And if it is not, the area under the curve gives you the p-values. And this is generally how statistical tests operate. They will calculate something called a T ratio or a F ratio from your data and look up a distribution and ask whether that value from your data is an outlier. And if it is, the p-value associated with it, the chance of seeing it will be very low. But there is another catch here, and that is pseudo-replication. So the distribution that is looked up, the null distribution, so to say, is dependent on the test that is used, as well as the number of samples you're using. And of course, that means the number of independent samples. So if you end up including technical replicates in your analysis, the wrong distribution will be looked up. And therefore, this will result in something called pseudo-replication, which means a false replicate. And this usually results in very low p-values. And you would end up thinking something is significant even when it is not. So remember not to include technical replicates in analysis. So what is the p-value then, and what is it not? So basically, if you assume a null hypothesis, and remember most tests, or all tests in fact, start with a null hypothesis. In a student's t-test, in the simplest case, the null hypothesis is the 2 group means are the same. So with that assumption, and with representative independent sampling, what is the chance of observing a test statistic at least as extreme as the one calculated from your data? By convention, if that chance is less than 0.05, we consider that as significant. But remember that this tells you nothing about the chance of reproducing the same result if the entire experiment is repeated by someone else. And this is why you need power analysis, so that someone else, when they repeat this, will also see the same small p-value if that is the true result. So with that in mind, we can move on to data visualization and the R package that I've created to make a lot of this a little bit easier. I want to just mention ggplot and associated family of packages in R. They're very, very common. They're very powerful and flexible. And they're highly, I would highly recommend people looking at how to use ggplot, for example. And in showing data, you also want to think about how you want to show the center of your data, which is usually the mean or the median. How do you want to show the dispersion? Do you want to show SD error bars, confidence intervals, violence, and so on? And of course, there are other things to consider, which I won't touch upon, but you'll be able to read this on some of the websites I've linked. So Graphify, it's basically a package which integrates both visualization and analysis. Because when I was doing this, I realized in R, you have packages that can either do plotting and data visualization or do analysis. But then I decided I wanted everything in one place. And so Graphify will let you do various kinds of plots, including bar graphs, box and whiskers. You can show distributions as violence. If you have before and after matched data, you can use some of these kinds of graphs. You can do time series. You can also do density or QQ plots or data distributions as histograms. So fairly large number of commonly used graph types. But why choose Graphify, or why did I make it in the first place? And one of the main reasons is after you do an experiment, you should be plotting it and just seeing whether the experiment is telling you something which you can just see by your eyes. Does it pass the eyeball test, sort of? And so this is the code for ggplot, just to plot a two-way ANOVA as a bar graph with mean and standard deviation in a bars. But you can just use Graphify with these 5 lines of code to get the same thing. So if you have lots of data to process quickly, Graphify makes this much easier and faster. The other thing is Graphify also offers more than 15 colorblind friendly palettes, which means they're all safe for people who may have different types of color blindness. They will still be able to appreciate differences that you want to predict or you want to depict with colors. The other thing I have also included in Graphify is mixed effects models. And I just want to show you, without getting into too much detail, what the benefits are of accounting for experiment-to-experiment error or inter-subject error, which means you use one batch of mice or one breed of mice, and then you repeat it with another batch of mice. You may want to take those inter-subject noise into account. So I've shown here, for example, with mixed ANOVA, this particular line here, the block represents experimental block. And this is the deviation associated with that. It's a factor that we are not interested in, but because we have managed to find the deviation associated with that, the residual deviation is far smaller. And therefore, these signal to noise ratios are relatively large and the p-values are small. But if the same data are analyzed by ordinary ANOVA, this standard deviation of the experiment and the residuals are now all in one place. So your denominators in these F ratios are large, and you end up with much smaller signal to noise ratios and larger p-values. So you can see that you have more power to detect differences using mixed models rather than ordinary ANOVAs. So when possible, use them. And last but not the least, I'm fully aware that a lot of people are not into R and can't do R yet, or may not be at a stage that they have the time to learn R. So just about a month or so back, I have made a new website, which is called Graphify Online. So if you use a browser and visit one of these sites, you will be able to do all of the stuff I was talking about that you can do in R. You can now just do it in a browser by just uploading your data in an Excel format, and then you can plot your graphs, do your analysis, download them, save them. You don't need any coding. But that if you ask me, if you're just starting out in science, it would be better for you to start learning R than starting out here. But this is accessible to a lot more people. So with that, I'm just going to come to the end and just summarize what I've been talking about. I hope you've seen how data analysis and data planning the experiment go hand in hand, because you can decide which comparisons and tests, and they talk to each other. You should decide how many independent samples to take, or how many repeats experiments to perform based on power analysis. This is typically important for high resource experiments such as with mice or human donors. I didn't get to talk about this, but it's also very important to record data in a clean and consistent manner, visualize them in as many ways as possible, and find the graph that conveys the message most clearly and in a most simple manner. And of course, we went through some of the linear model methods where you have to diagnose the residuals, then perform the planned experiments, and then report all of your methods and data handling procedures in your method section. So with that, I'm going to stop. This is the link where you will find all the Graphify and the stats resources, as well as the R workshop. And I just want to thank colleagues from the Department of Computing for helping put together one of the 2 Graphify servers online. And I hope you try it out and give me some feedback on that as well. And with that, I just want to thank everyone for your attention, and I'm happy to take any questions.
Okay, am I audible? Yeah. Thank you so much. That was a very insightful session, and I'm sure that all of us have gained a lot of insight from it, because there are many common mistakes which we make
while processing biological data. Like you said, doing the experiment is easy, but the analysis is the tough part. So I'm sure everyone benefited a lot from it. So we have some questions in the Q&A box. The first is, do we have to memorize all this code when using R? I am not from computer science background. I tried to use R. It is easy, but I find it hard for me to memorize the code.
Yeah, no, that's a great question. No, you do not need to memorize the code, and I don't memorize the code either. And you'd be surprised how many times I just Google or go to Stack Exchange to find how to do a certain bit of analysis. But the benefit of R is once you've written it down in an R markdown file, you have that code and you can just keep repeatedly using it with your new data file and just keep running it. So typically you end up needing to just write it once and then edit it.
Okay. Okay, the second question is the Graphify package accessible inside the R environment?
Yes, it is available for free to download from CRAN or GitHub. So if you're an R user, you should be able to just download it and install it. The website shows you what other packages you might need to run it, because it depends on ggplot, obviously, and a couple of others. But yes, it's fully available within R.
And what do you think about the Gen AI like ChatGPT to do the coding in R?
That's a great question. I have started using it, and I did use it fairly to make what is called a Shiny app. The browser-based version of Graphify is a Shiny app, which is another package to make websites. And I did use Copilot, which is a coding-based AI, and it's useful in some bits, but it does make a lot of mistakes. And unless you know the language, there's no way you can fix those mistakes. So learning the language there's no option but to do it. It's just that if it's some base code that is boring to write, you can get a skeletal code, but there is no guarantee it's going to work, and most often it doesn't.
Thank you. Like we don't have time to take any more question and answer right now, but you can like whoever has asked the questions, you can mail us in the IUBMB Gmail address, and we will get back to you on the questions. Also, there was a request, Dr. Avinash, to share the book which you said in the first slide. If you could show that again and the resources, because I think multiple people asked about that in the chat box.
Sure. This is the one. If you go to shenoylab.com, all of the links can be navigated from there.
Okay. Thank you so much, Dr.
You're
Avinash,
welcome.
for
Thank you so
patiently
much.
answering
Thank you
all.
very much.
Yeah. Okay. So, without further delay, let me introduce you to our next speaker, Dr. Chan. Dr. Chan is a senior principal research scientist from Duke-NUS Singapore. Dr. Chan has been integrating multi-omics datasets to provide molecular insights into viral pathogenesis, virus-host interactions, and the vaccine correlates of safety and protection. Dr. Chan is going to showcase to us how his team have used Python to understand host responses to vaccines and how Python can be eventually used to develop web tools for biomedical research. Let us welcome Dr. Chan. Over to you.
Thank you. Yes. First of all, I would like to thank the organizers for inviting me here. I think it's a great opportunity for me as well, as of course to all the participants as well, to share some of our knowledge that we have gained over these years. Trying to so now with all the big data, so how do we use big data and how do we interpret big data to get biological insights? Usually, at least for now, it seems to be the bottleneck rather than the programming language itself. So without further ado, but before I go into the topic, I just wanted to say that this is not about comparing, whether R is better or whether Python is better. But at least I hope that after this session, it convinces some of you at least to try to learn the programming language, whether it's R, whether it's Python, because they are really important in terms of data analysis. And I think at least learning some of the foundations will bring you a really long way in your career, even if you know just a little bit. Yeah. So obviously you can learn a lot of things online, and with ChatGPT, you can ask a lot of questions. So I will not reiterate what's published online, but rather share with you some of our experience that we have. So actually, for me, I'm not a computational science major. I started out as a wet lab researcher, and then transited towards a dry lab. So hoping that my session here will also convince some of you that you can actually do the same as well, and you are actually not lagging behind just because you didn't start out as a dry lab researcher during your undergrad or postgrad. So who am I, right? Because I do a lot of omics data analysis as well as single cell data analysis, a lot of people think that I'm actually from a computational science background major, but actually that's not really the case. I started out in my PhD, and part of my postdoc is also looking at mostly at cell biology, trying to understand what are the receptors that are involved in dengue virus infection. So actually, a lot of my first few papers are actually mostly 100% wet lab researcher. But over the years, because I had more exposure to human data sets, which is very heterogeneous, I had to learn the programming language, and this allowed me to transit towards a dry lab where now I run a lot of clinical trials and analyze clinical trial data to try to understand why different people respond to vaccines and infections differently. So I also want to highlight that, of course, if you start out with a computational science major, there's some advantage because you have a good knowledge of the how the programming language work. It allows you to develop a lot of software and methodology related to computational science. But I also want to highlight that it doesn't mean that if you start out with a wet lab, then you shouldn't transit towards dry lab. There is also some advantage. The main advantage I can see here is that you will have the domain knowledge, which allows you to apply your bioinformatics that you have learned during your dry lab and apply the language to understand what is happening behind the biology. Yeah, so here I'm trying to convince all of you that it's never too late to learn bioinformatics, and actually learning some of them will be quite useful in terms of statistical analysis, as what the previous speaker has already highlighted, and even some of the machine learning algorithms that will come useful along your way. So the question often is, how do you really learn anything, right? Of course, as you can see in this picture, right? Usually if you want to learn anything, the social aspect is quite important. That means if everyone is learning the same thing as you can progress quite fast because you have a community that can actually help you. So that's one way for you to learn a programming language, for example. Of course, the other thing you can think about is to find a shorter hill. Yeah, so hoping that you pick up a language that's probably easier to learn. And I think if you are thinking of an easier to learn programming language, it's definitely a Python. Yeah. I thought I would share with you, some of the timelines of when R or Python was developed. So R was actually first developed in the 1970s. Basically developed for two primary reasons. One is data visualization and statistical analysis. Yeah, so as you can tell, a lot of the R packages actually do all of these statistical analysis as well as data visualization very well, even much better than Python. But in the 1980s, actually some people think that the R programming language was very difficult to learn, so they actually developed Python in a hope to make the language easier to understand, and thus is a more elegant language to learn. Yeah, so if you are hoping to learn something new, I think and you are quite intimidated by the language, Python is often an easier platform to learn compared to R. And that was why it was developed in the 1980s. But what was also interesting is that the language actually took a slightly different trajectory compared to R, in that a lot of the machine learning packages are started to be developed in Python, including a lot of the machine learning, deep learning, and artificial intelligence are actually developed in Python much more than R. So if you are thinking of machine learning and AI, definitely Python is a language that you cannot escape. Yeah. So this is just a rough chart of why is gaining popularity. Is particularly because Python is much easier to learn as compared to R. But of course, a lot of the science requires R because of its very good statistical tools. So in the best case scenario, you should learn both languages because they kind of do different things. But if you are just using as a platform or as a tool for data analysis or machine learning, then definitely Python has some advantage compared to R. And that's also one of the reasons why many of the careers are actually looking for people who at least know how to do some Python programming. So I thought I will share with you some use for Python and why you should consider Python, especially when you deal with the big data sets. Yeah. So one of the big aspect about using Python is really the machine learning and the deep learning part with Python. And why is so much better is because they have one of the most comprehensive packages that is made for machine learning and AI. Some of these includes TensorFlow, Keras, SciPy is also very popular nowadays, and also sklearn, which has a very good network of people using. So in terms of troubleshooting everything, there's also a big community that can actually help you troubleshoot a lot of these questions that you may have when you're using machine learning or AI. So just a short note of what is machine learning and why it may be useful. So in a lot of the omics data analysis, we are measuring like 10,000 different transcripts or genes, right? Or thousands of different metabolites. So it's very hard to analyze one by one and be able to tell what is important and what is not. So the hope of machine learning is that instead of looking at 10,000 genes, for example, you are looking for patterns, right? Maybe you identify 10 groups of genes that follow similar patterns. So rather than analyzing as 10,000 genes, you are now looking at 10 different groups. And then based on this group, then you can correlate with a phenotype, for example, to see, whether there is significant association or not. So that's what machine learning tries to do. Hoping to reduce the dimensionality of the data so that you can focus on what matters and help you make a more informed decision of your predictions in terms of from your data set. So we just to showcase one of the machine learning algorithms that we have used to predict vaccine responses. So this was a clinical trial that we have initiated in Singapore during the COVID pandemic, trying to develop a vaccine against COVID-19 disease. So we recruited about 106 subjects for our clinical trial, of which 78 are vaccinated and 28 are placebos. The idea here is to understand what is the host response that is associated with antibody response that we know is important for vaccine-mediated protection against the virus. So what the machine learning does is that at least based on the random forest algorithm, which is one of the most popular machine learning algorithm for predictions. So what it does is that based on say 100 subjects, we take 80% of the data as the training data. And what we do is based on this 80% of the data, build a decision tree to understand how we can separate the high antibody response from the low antibody response. So here we build one tree. And then what we do is that then we repeat the process again, pick another 80 subjects randomly, and then build another tree. So we are building this tree 1,000 times, and then take an average result to make a prediction. Yeah, so all of this is actually very doable in Python, and impossible to do it manually. So that's what the power of the language does. So by building this tree, we identify 6 genes that are predictive of what the antibody response is for the vaccine. And based on these 6, we can see that the prediction is very good. Yeah. So rather than looking at which gene is associated with antibody response, now you are looking at 6 genes which more accurately predict what the antibody response is, which is not possible to do it if you are doing Excel, for example. Yeah. So that's the power of one of the utility of machine learning, which allows you to understand or to reduce the dimension to allow you to focus on what response matters and use what the data is to infer the antibody response in this case. So we were also rather interested to find out what are the machine learning models that are also most applicable in terms of infectious disease research. We look at public health surveillance, diagnosis, and prognosis. This paper just came out earlier this week, and then we found that a lot of these machine learning tools, including explainable AI, ensemble methods, which includes the random forest, which I described just now, and neural networks are actually very prevalent in terms of infectious disease research. And all of them can be actually easily performed with the Python programming language if you understand the language. Yeah, so if you are into machine learning and big data, I think Python is something that you may want to be familiar with if you want to develop a future career with machine learning. Here, I also want to highlight that, of course, random forest is one of the machine learning tools they can use, but actually there are also many other machine learning algorithms that can be employed. So an advantage of learning many machine learning algorithms is that it allows you to see the data in many different ways. So allowing you to see in various perspectives of your data. Then based on your perspective of data and also what is known from scientific literature, you can actually make a better decision based on how to interpret your data or your big data that you have generated from your experiments, for example. The second utility other than machine learning is that Python also allows you to curate data very efficiently. And the powerful thing is after curating the data, you can further develop it into a web tool that allows other researchers to analyze what you have curated as well, making it a very useful public resource for future scientists. So why do we bother about data curation? Is because humans are generally very complex. Everyone has different genetics background, have different environmental exposures, have different microbiome, and even different other sleeping habits and stuff, which can affect any kind of responses, whether it's response to vaccines, whether it's response to a therapy or response to infection, for example, right? So sometimes you can use this heterogeneity to your advantage, in which you can understand why different people may respond to a vaccine or a virus differently. But sometimes this heterogeneity can be quite complicated. So So you may want to untangle what is signal as compared to what is noise. So that's the difficult part. But the one way I think you can improve the signal to noise ratio is that you can increase the sample size. And how you can do this is by integrating multiple data sets from various data sources, especially now that there are a lot of publicly available data sets that researchers are uploading. So what you can do is, in addition to what you are doing, you can integrate data from public data sources, and then make a more informed decision of what is really happening. And that's something that I think the Python programming language can do quite well. Yeah. So this is also one of our study that we use meta-analysis to try to understand what are the key gene expression differences that you see in severe COVID patients. So here we actually analyze 7 data sets to identify 2 genes that are most important for prognosis of severe COVID. So this is the value of doing various data sets, and this allows you to decipher what is a signal and what is noise if public data base are accessible. And then what we do is based on public data and based on our own data sets, we can make combine all of these data to understand what the biology and what is the biomarker that is most suitable for severe COVID prognosis, for example. And of course, you can also use the database. You know, if, for example, if you are using, you are developing a new vaccine, you can actually use the database to see whether your vaccine, how does your vaccine response affect with respect to other vaccines as well. So we have a lot of database with other vaccines, and we can see what kind of host response they are similar with other vaccines and what host response are different. And finally, I think Python is very useful nowadays, especially with the recent development of Streamlit, which is freely available and very easy to execute. So actually, all of us can now actually make web tools rather easily, especially with this Streamlit. And the good thing about Streamlit is that you don't need to pay, and and it's very easy to implement. Yeah, and it's publicly available. So if you're interested, you can check this website. We have also used this Streamlit to make multiple packages as well for clinicians and other researchers who do not want to venture into programming. So one example is, for example, we made this omics data analysis tool that allows users to actually just upload the data. For example, gene expression data or protein data. You can actually do multiple different kind of analysis using a web tool interface. For example, you can plot volcano plots, you can plot differential analysis, and everything will be updated live, depending on what kind of threshold that you're setting and what kind of yeah. So it's made so that researchers can quickly upload data and have a sensing of how their data looks like before doing further analysis. So this is just an example output. So for example, we put in one of the data sets, and you can quickly see, at day 1, you have a good expression values, and then based on volcano plot, you kind of know what are the top genes to look at, and you can also look at, for example, their functions of the genes and what pathways are they involved in very quickly within minutes. And this is not the only web tool we developed, but we also develop other tools as well. So for example, we developed one tool which actually can allows easy implementation from Excel into say Python or R very easily. The other one is a heat map plotter, which you can just upload your data, and then you can set your own threshold and colors, allowing you to generate a publication quality figures that can be easily downloaded for publication. And finally, if you have databases, you can actually put multiple databases into your database, and then it becomes a web tool that allows other researchers to actually look, for example, their gene of interest, to see how their gene is changed with time, for example, or with different conditions. So all of this can be easily executed and very easy to maintain as well. So if you're interested in making some of these web tools, you can visit my GitHub. All of these are actually publicly available. You can easily download them, and then you can run them locally. And then if you want to tailor make to whatever your applications are, you can just change some of these commands, and you can eventually make your own web tool locally, or you can, if you want, you can also make it publicly available. Yeah. And finally, just to end off, why you may want to start with Python is because the hill is a little bit shorter compared to other programming languages. So it may be less intimidating to learn. But having said that, I must say that both R and Python are actually very important in terms of big data analysis, and I use R a lot as well. But if you're new and you're starting out, I would recommend Python. Yeah, definitely. And finally, just a simple acknowledgement slide to my team, comprising of postdoc, RAs, and students as well. Thank you.
Thank you, Dr. Chan, for that great presentation. It's now time to open the floor to some questions, and we'll try to get through as many of these from the Q&A box as we can. The first question we've got, which is always a good one to get started with, is we want to know what a good source to learn Python for a beginner would be.
So how I learned was so it depends on what kind of learner you are. If you enjoy reading books, they are very good books. They are cheap and easy to read. But for me, I think learning programming from books is really difficult. So what I do is I use some of the tools that other people have developed. For example, Data Quest. So it allows you to have so it gives you questions to answer. So then you have to input your answer to answer the question. So to me, this kind of learning is a bit faster. So it feels like I have to solve problems to advance my learning. Yeah. So for me, I recommend Data Quest. And I think a lot of people also benefited. Although it's a paid service and I don't get any royalties from it, but I've benefited a lot from Data Quest. Yeah.
We have another question. Could you please compare the parallel processing in Python and R for large data sets?
Oh, yeah, that's hard. I think R is a bit more efficient in terms of running a lot of the programs. I think much more efficient than Python in many aspects. I feel like many of the processes that are run in R are actually executed much faster. Yeah, but in terms of machine learning, I find R very difficult to implement. So that's especially if you are using machine learning tools that were developed in Python. I think they are very difficult to implement R. So if you are really into machine learning, I think Python is definitely a better language although it's a bit slower, but I think it's still much worth to invest in Python. Yeah.
We have a question about data cleaning in Python and how data cleaning works in Python.
Yeah. So data cleaning in Python and R, I think they are quite similar. I think a lot of the syntaxes are quite. I mean, there are some differences, but I think many of them, the thinking of cleaning and wrangling the data is quite similar. For example, like pivot tables and also how to remove how to remove columns and stuff. So the logic if you understand Python, you will understand R as well, and vice versa. Yeah. So I think both are equally easy to learn. But I started out with Python, and I think it's quite easy to clean the data with Python. Yeah, because you can just once you have developed the pipeline, you can just easily execute the same pipeline for any data set. Yeah.
Another question. So we've got a beginning person who's starting off in data science, and they'd like to know any core skills that you think are essential for a biological scientist to master early on with Python.
Definitely. I think the number one thing you may really want to be very good at is definitely data cleaning. Because I think a lot of time is actually, I would say, close to 70 to 80% of the time is actually spent on data cleaning. In terms of making sure that your quality is good and you remove all the data that has missing values and stuff. And then the other 20% is really, to implement the machine learning and all the for example, comparison, statistical analysis. Yeah. So I think if you want to be in this field, I think you will want to, one, understand how to do data cleaning. Two is understand the statistics and the assumptions behind every library or statistical comparisons you are doing, or even machine learning as well. I think that's very fundamental to you implementing or gaining insights from this kind of big data analysis. And then the third thing is at least have a sensing of what kind of packages to use. Yeah. I personally don't think that you need to know every single machine learning algorithm to be good. Because every data set is different, but at least you need to understand the assumptions and the statistics behind them. I think they are quite crucial. Yeah.
Okay, I think we have time for one last quick question. So someone has asked if you could do a Python analysis using machine learning packages just on a laptop, or whether you would need access to a more powerful computer.
Oh, you may be surprised. Actually, most of the Python analysis can be done on a laptop. Yeah. So actually, that's also the advantage of being in a dry lab as well. So essentially, you can work even from home. But of course, the more upstream ones, for example, like in RNA-seq or single-cell-seq, where you have the very raw data sets where you need to map the genes and stuff, that will require a more complicated supercomputer to help you analyze. But after you process the data, everything can be done on a laptop. Yeah, no issues at all.
Amazing. Thank you very much for your presentation, Dr. Chan. And we just have a couple of final slides to wrap up. Just share my screen. Oh. Cool. So a massive thank you to Dr. Avinash and Dr. Chan for sharing your very valuable insights with us today about R and Python. We really thank you for your time and for joining us today. The IUBMB has also been organising many other webinars and workshops over the years. To see some of these previously recorded sessions, such as the Scientific Illustrations session using Blender by the American team, you can scan the QR code on the right here. Please follow us on social media to learn more about the opportunities and activities we offer. We're on Instagram, X, LinkedIn, and Blue Sky. And if you scan this big QR code here, you'll be able to join our mailing list. Mā te wā and farewell for now, and we hope to see you at one of our future events. Thank you, everybody.